Good afternoon and welcome to the final Retina Australia webinar for 2024, Cell Therapy, Genetic Research and a Patient's Perspective. I'd like to begin by acknowledging the traditional owners and custodians of the land on which we meet today, the peoples of the Kulin Nation. I also pay my respects to their elders past and present. My name is Sally Turnbull. I'm the Administrative Officer for Retina Australia and I will be the facilitator of this webinar today. As a participant, your camera and microphone will be switched off throughout. There will be an opportunity for you to ask questions once all our speakers have finished their presentations. If you have any queries during the proceedings, you can type a question to me in the Q&A section. Our first speaker this afternoon is Associate Professor Anai Gonzalez-Cordero. Associate Professor Gonzalez-Cordero is an Al and Val Rosenstrauss Fellow and a group leader at Children's Medical Research Institute at the University of Sydney. Associate Professor Gonzalez-Cordero is a leader in the field of stem cells and their differentiation into organoids, with an emphasis on translational research to develop novel therapies for retinal genetic diseases. In her presentation today, Associate Professor Gonzalez-Cordero will present an overview of her research on developing cell therapy to develop a universal therapy for inherited retinal blindness. She will explain how stem cell technology, including retinal organoids derived from pluripotent stem cells, serve as an ideal source of cells, specifically the light-sensing cells of the eye, the photoreceptors for transplantation. Associate Professor Gonzalez-Cordero will cover the pathway towards developing this advanced therapy for the retinal prosthesis and how they can improve the functionality of the devices.
Hi, good afternoon. It's my pleasure to be here presenting a little bit of the work that we do in the lab to you. So this is part of our research. Last year, I know that Vivienne Kaiser presented on our work on gene therapy. And this year I want to concentrate on our work on cell therapy. And I hope my sound is clear. And thank you for the introduction, Sally. So as Sally mentioned, we use stem cell technology. So these amazing cells that can generate all the cell types in your body to develop cell therapy to treat blindness. And I'll start my talk. I always like to have the team there because I never do any of this research alone. They are the ones doing the hard work every day in the lab. So I like to acknowledge the work. And just a brief introduction of where we are and where I came from so that we set the scene. So I actually did my degree at University College London, Institute of Ophthalmology. And I moved to Sydney in 2019 to establish my lab at the Children's Medical Research Institute. And apart from my lab, I also lead a stem cell and organoid facility. So we are all about stem cell technology in the lab. And of course, my group focuses in inherited retinal diseases. But we like to offer this technology to all researchers around us. And I'm also associated with the University of Sydney. I'm associate professor there. So I'll start with the stem cells. Again, I'm sure Vivienne introduced these cells in the past, but just to set the scene, what are pluripotent stem cells? We like to have the analogy of baby cells in the lab because these cells actually come from a very early embryo. So the blastocyst stage, when after fertilization, the egg starts to develop, we have these amazing cells, as I like to always refer to them, that can give rise to a lot of different cell types. And in this knowledge, we talk about a cell that wants to grow up to become a baby, that wants to grow up and become any cell type, but the analogy is that they are instructed and educated to become the different professionals, an accountant, a nurse, a barrister, and so on. So, of course, in our body, what happens is that these unspecialized cells that like to divide and grow and renew themselves and become just another stem cell. During the period of development, they are instructed by signals in our body as the body develops to become a heart cell, a muscle, a neuron, or a kidney and so on. So they have this potential of going from a undifferentiated cell, so which it is a unspecialized cell, into a very specialized cell and forming the many cell types that we have in our body. So as I mentioned, just to recap, an embryonic stem cell is a cell that came from a very young embryo. We can isolate this cell, so it's depicted here by this yellow mass that can be then grown in the dish in the lab. And here is just an image of what we usually see under the microscope. So in the embryo, when these cells start to differentiate and become the cell types, this is the process that we call differentiation. And here it's, again, just to really emphasize this. And when I'm talking about differentiation, you understand that it's this process of the stem cell becoming now a differentiated cell type of your body. So, of course, embryonic stem cells were discovered quite in the 80s. These were mouse embryonic stem cells that were isolated from the blastocyst. And again, we can grow them in the dish and form any cell type. Nowadays, we know how to instruct these cells to develop many organs. Here, I'm just showing some very early neurons and then how they can mature to form quite actually developed neurons. And this was mouse, but of course this technology was then translated into humans, and we were able to generate these cell lines that can be banked and used in research. Now, back in 2012, we had a breakthrough in stem cell research, because Shinya Yamanaka, a Japanese researcher, discovered the induced pluripotent stem cells. And he actually won the Nobel Prize in medicine for this discovery. And what he did was to actually turn the adult cells, so let's say your blood cells, your skin cells that already have been through that process of differentiation, he isolate those cells from individuals, put them in the dish, and he tested a number of factors on these cells, and factors that are involved, that we know are involved in the development of the stem cell. So to be a stem cell, you need to have some genes turned on in that stem cell. He basically isolate those genes, he put it back inside the skin cell, and that skin cell, and he found the ideal cocktail to bring that skin cell now back in time to be our baby cell. And that process, and that is depicted here. So for example, now we had all our different adult cells, our different professions, and with 4 different growth factors, transcription factors that he found that are expressed in the stem cell, now these cells can become baby cells again. And this process of turning back in time and inducing this process of becoming a baby, a stem cell again, is called, it's a process of induction. So he called these induced pluripotent stem cells, or iPSCs for short. So if you hear me talking about iPSCs, is because now these cells came from adult cell that became a stem cell. And why is this important? It's important because now we can take a cell, a blood or a skin cell from a patient, and then make new stem cells from these individuals. We can make in the lab stem cells from anyone, really. And why is that important? Because as I told you before, the cells grow, they proliferate, and we know in the lab how to make then the stem cells into other cell types. So what are the advantages of iPSCs, of having these cells? So now we don't need to use embryos anymore. We can take these cells from any patients with diseases, children's, patients with different genetic makeup. So we have a repertoire of cells that we can use in the lab now. And of course, because we can get these cells from individual patients or or family members, for example, you can do personalized medicine. So you can have person-specific iPSCs that can be then differentiated into a kidney, a liver, an eye, a brain. It depends on what we want to study in the lab. So just to recap once again, so we can have a patient with a confirmed diagnosis for inherited retinal diseases, let's say, and we obtain the blood. We like to work with blood in the lab because it's less invasive, easy to get. And we bring it back to the lab, make the iPSCs, the induced pluripotent stem cells, and now we can then start that process of differentiating, so generating the eye cells, in this case, these beautiful retinal-like organs that we call organoids in the lab. And then once we have these organoids growing, which mimic the environment of the retina, we can have thousands and thousands of these organoids growing to investigate the diseases, and we can also test new therapies. So it's very exciting for us to have this material. Okay, so a brief background on stem cells, and let's move on to what we actually do in the lab to develop therapies. So we can grow these organoids, and here I'm just showing, I like this picture because it shows in green your cone cells, human cone cells that we generated in the lab. And you can really see the lamination, the beautiful structure of the cone cells that we generate from stem cells. They're very typical cone, like they're a bit chubby, and they have these huge pedicles that actually form synapses. And I'm showing here the spuntated markings are synaptic proteins that we can see. So we can generate quite a lot of photoreceptor cells, which are the cells that are necessary for vision, as you know. So how do we do that? We do it by making these mini organs, the organoids, and it's basically putting together a bunch of those baby cells, the embryonic stem cells, the iPSCs, and they actually do a very good job of instructing each other to become an eye cells or a mini retina. Of course, in the lab, we do provide them with cocktails of media, of proteins and supplements every day that will help them decide, okay, instead of becoming a brain, I'm going to become an eye. And this is what we have expertise on. And they are called organoids because they grow in 3 dimensions. So they grow in suspension in the dish, and they really form a structure that it's reminiscent of the retina. So we now have these mini human retinas growing in the lab. And the program of research that I do in the lab encompasses many questions, but all centered around the stem cell technology. As I said before, we do a lot of disease modeling and gene therapy. We also engineering organoids. We want to understand human eye development in the dish now that we have these organoids. But today I'm going to talk about cell therapy. So why cell therapy? So as you know, inherited retinal degenerations due to the loss of photoreceptor cells, it's one of the main causes of blindness in the development world. And these are mainly caused by retinitis pigmentosa and maculopathy diseases. In retinitis pigmentosa, for example, you are losing the rod cells in the periphery of your vision. And in maculopathies, you're losing your central vision, your color vision, because of defects in the cone cells, in the macula, in the central part of your eye. Of course, there's other conditions. I'm simplifying this, but at the end of the day, all these conditions are caused by the loss of your photoreceptor cells, so the light sensing cells of the eye in the back of the retina. What we do, so when one think about a treatment for these conditions, you need to really think about the stage of disease that you're going to treat patients, hopefully. So you have your healthy retina with all the laminated retina, beautiful photoreceptor cells. When you have early to mid-stage disease, these cells start to die. So we start to lose a few of these cells, but you still have some cells remaining there. And when you have advanced disease, you only have very few, and usually they don't look very good. Your inner retina, the other neurons that the photoreceptors connect with, are also starting to suffer at this stage. So not many photoreceptors remaining in this case. So if you think about therapy, and how we do our preclinical testing of these therapies, you need to think about the stage. So early to mid stage, where your photoreceptor cells are still there, one can think of receiving perhaps drugs to slow down the progression of disease, or you can deliver gene therapy, which is basically replacing the gene that is faulty on these cells. And that's because the gene therapy needs a cell to act upon, and you still have some photoreceptors there, and the same with the drugs. But once you lost all your cells, and these cells don't regenerate in the human eye, one can think of a treatment, for example, the electronic array, what we call the bionic eye, which is an electronic chip that is implanted at the back of the eye and helps to actually rescue some light sensitivity in patients. And it's in clinical trials, and there's some of these available. But in the lab, really what we do is to try to develop a more physiological manner of treating, of offering therapies to these advanced stages of disease. So we basically make new photoreceptor cells using stem cells, and we want to then replace these cells. And we think that this replacement of photoreceptor cells, it's viable because we can actually now obtain induced pluripotent stem cells from a healthy individual. We can make the stem cells. We can then bank these stem cells and have a bank of cells to generate healthy retinal cells. So millions of photoreceptor cells that can also be banked. And then we have cells pretty much on a vial to be transplanted into patients. Of course, this is the end goal. This is where we want to be in a few years, what we want to achieve. But the principle, we believe that this is a good approach because, again, when you're losing these cells, all you need to do and I say all you need to do a very in brackets, because it's actually more complicated than it sounds. But if you replace these cells, they have to do a single synaptic connections with the other neurons of your retina, because this is a sensory neuron. So on the other side, you're going to have the light sensing part of your cell receiving the light, and this needs to be transmitted through synapses. And of course, the eye is accessible, so it's quite easy to do the surgery. Again, with a pinch of salt, because it's a very precise delivery of cells or gene therapies at the back of your eye. So we say it's easy when you compare with a brain surgery, for example, but it's a very delicate surgery. And it's a relatively simple structure. So you lost your photoreceptor cells, and you can they have to survive and connect and mature. So I think the things that I like people to remember are these cells that we transplant, they need to be able to sense light. Of course, they need to be function, and to be function, they need to have these specialized cilia here, the outer segments. And they also need to connect with the other interneurons, so form synaptic connections so that the electrical impulse can be transmitted all the way to the brain. So I have been doing cell therapy for quite a while now. I started this work in London. They had already done quite a lot of work on transplanting mouse cells into blind mice. I then did my PhD there to transplant mouse embryonic stem cell derived photoreceptor cells into blind mice. And then later on, I moved on into making human cells. And I spoiled here that we actually were able to show visual restoration, which is quite exciting and allowed us to be thinking about moving this technology to the clinic. So I'll just touch on that a little bit in this presentation. But before I show the data, I need to explain briefly how we we actually make these cells. This is a complicated slide just showing that we can make these organoids from stem cells. This is our pathway. And it's not a trivial task, I have to say, because we actually have to grow these cells for 15, 17 weeks in the lab before we have photoreceptor cells at this stage, at the correct stage for transplantation. So one of the first studies that we did was to actually find out what stage these cells are the best to be able to connect and mature, as I said, the two requisites of a good cell transplantation. And we have found out that is around 17 weeks. So there's no escape. We need to culture these cells for that long. And of course, this is an expensive task. And when I developed this protocol back in 2017, I had that in mind. You know, we need something that is relatively easy for the researchers to do. This is a lot of work in the lab, a lot of manual work. We need something that is affordable because all this research is very expensive. So our protocol took all of that in consideration. And we have a beautiful protocol that actually generates thousands of these organoids. And these organoids have all the retinal cell types. Actually, because it's modeling the retina. But remember, we want to transplant just the photoreceptor cells. And what we do is to isolate. So usually we mark these cells, the photoreceptor cells, with a viral vector. So we use gene therapy in these organoids to just mark these cells in green with a jellyfish protein called GFP, green fluorescent protein. We are able to then isolate these cells out of the organoids and transplant. And this is a section, a cross-section of an eye of a blind animal. And it had no endogenous photoreceptors left. So there were no mouse photoreceptors, very advanced stage disease. And hopefully here you can see all the green cells that we have replaced with in this mice. So we transplant 500,000 cells into these animals. And what we see, again, very briefly, just this has all been published and it's a beautiful story, actually, if you want to read. But what we do then after transplantation is to let these cells mature in the eye for another 3 months. And we do behavior studies. So here is a simple study where we have a chamber. So with 2 compartments, one with light and the other one is dark. Mice are nocturnal animals and they have preference for the dark compartment, so they feel more secure in the dark. And the blind animals don't have, which I'm showing here in dark circles, dark circles, black circles. They don't have a preference for the light or the dark because they can't sense light. So they spend 50% of their time in both compartments. Now, the animal, the wild-type animals that can see spend the majority of the time in the dark. The animals that have received the healthy cells, the green cells in this case, now are spending significant more time in the dark compartment. And we also did some shunt transplantations with photoreceptor cells that came from patients with CNGB3 disease. So these are cone cells that are defective, so they cannot sense light. So our perfect controls in this case. And these shunt injected mice could not perceive, and they also had no preference. So it's a relatively easy way to show that these animals after transplantation are actually perceiving light and now choosing to spend more time in the dark. And we can track their paths and film their activity to really show their preference. So again, this was really remarkable for us. And what we are doing now is to improving upon this technology. And I'm not going to show the data of what we're doing to improve transplantation, but rather just describing the last couple of minutes of my presentation, how we're moving this technology to the clinics. We have a large MRFF grant on translating everything that we do in the normal lab, which is a standard laboratory that it's very clean and nice, and we try and be as organized as possible, but it's no mean a lab where we could produce cells to inject in humans. So what we want to do now is to translate all that we do in the lab to a good manufacturing practice facility, so that it's a facility that is going to manufacture the cells for us in a compatible, that so that the cells can then be used in humans. But before we get there, what we need to do is to find an iPS cell line, so our baby cells that can be that are also GMP and can be used in humans in the future. So that work is being done in Melbourne together with NAR from the a cord blood bank there, and we now have a couple of cell lines that we are testing in our lab. So it is very exciting that we have the first GMP iPS cell lines in Australia to work with. In my lab at CMRI, what we're doing is to translating that protocol that I described to you very briefly into GMP. So what we're doing is removing any animal materials, for example, checking every single ingredient that we use on a daily basis to see if that material can be used in humans. And if not, we need to find something else to replace. We're developing quality controls. We have all the documentation in place and so on. It's a lot of work. And in parallel, we're also developing psychosocial studies because we want to engage with the consumer to really understand what is their priorities in terms of inherited retinal diseases in general. What is it that they want from the researchers, the clinicians? What do they need? And then also we are just starting a new project where we want to understand the impressions on stem cell technology and in particular cell therapies. And that has been done together with the researchers at UNSW. So again, as I explained, we have lots of stages in this manufacturing so that we can hopefully one day isolate these cells to transplant them to move them into clinical trials. And I always like to emphasize that this is a field that it's under construction. There's lots of researchers doing it around the world. We have a group in the US. We have a group in Japan that in fact has already transplanted 2 patients with pieces of retinal organoids. We have the group in the UK where I used to work with that it's also trying to develop GMP manufacturing and moving this technology into clinical trials. And I very proud to say that we are the group here doing it in Australia and bringing this technology down here, because I think the patients here need to have access to these clinical trials in the future. And here just how we are actually thinking about introducing these cells, making enough of these cells. Just a little part of this project is to actually use robots to manufacture the cells. As I said to you at the beginning, it's a 17 weeks process, and to do it in a way that we generate enough cells, we need automation, and we have just installed these robots in the lab. So it's very exciting to see it happening now. And I think I ran out of time, so I'm just going to say thank you to all, and I think I'll answer questions later on.
Thank you so much, Associate Professor Gonzalez-Cordero. That's fantastic. It's so exciting to see how far it's coming, and I want to just hear more about it. But we'll now move on to our second speaker, and we'll have time for questions with you after we've had all the speakers. Our second speaker this afternoon is Dr. Ceecee Britten-Jones, a research fellow at the University of Melbourne and Centre for Eye Research Australia. She's also a registered optometrist and practices at the Australian College of Optometry. Ceecee's research focuses on improving the understanding of different inherited retinal diseases and improving support for individuals throughout the diagnostic process. Dr. Britten-Jones leads genetic research studies at the Centre for Eye Research Australia and the University of Melbourne, working alongside the team on the Victorian evolution of IRD's Natural History Registry venture. In this presentation, she will provide an overview of the new genetic research initiatives her team is leading and their impact on advancing IRD research and access to therapies. Ceecee will also talk about the current state of genetic research in IRDs, the processes required to achieve a diagnosis, and their team's collaborative efforts to improve outcomes for individuals living with IRDs. Thanks so much, Ceecee.
Thank you very much for the kind introduction, Sally. Thank you for having me, and I'm really excited to talk about some of our genetic research studies in inherited retinal diseases. What I'm really going to take you through in the next 15 minutes is not only some of the work that we're doing, but really talk you through the steps of how we actually do our work, what kind of data we generate, and how we go about trying to find new diagnoses and new mechanisms in inherited retinal diseases. And before I start, I will mention that I do have some slides, but really they're quite basic and they're really a visual cue so that I know and I remember what my talking points are. And so if you can't see the slides or if you prefer to just listen while you do other things, that's absolutely fine because I will sort of talk through what is on my slide as I go through. And so most of us hopefully all know that inherited retinal diseases can be caused by over 300 different genes. And that's just what we know of so far. And what we know is that the changes in these genes, they're like spelling mistakes. And these spelling mistakes are what severely damage the protein function that then leads to degeneration of the rods and cones that I have just mentioned earlier. And what this means for each affected person is that generally, most inherited retinal diseases can be attributed to be caused by changes in a single gene. And that's what we call a genetic diagnosis, right? But one of the challenges we have with IRDs is that there's a lot of different overlapping clinical presentations. And so what I'm showing currently on my slide is a bubble plot. And what we can see is a lot of big bubbles, and the big bubbles each represent a different clinical diagnosis. So for example, RP, retinitis pigmentosa, Stargardt disease, cone rod dystrophies, etc. Within each of the big bubbles, there's a representation of all of the different genes, or not all of them, most of the different genes that might cause some of these clinical diagnoses. And what we can see from this graph is that there's a lot of overlap between the big bubbles. And what this tells us is that in inherited retinal diseases, sometimes the same genes can cause multiple different phenotypes or clinical diagnoses. And the same phenotype on the other end can be caused by multiple different genes. And what this means is that sometimes diagnosis can be difficult, and getting an accurate and complete diagnosis can be challenging without the specific genetic information. Now, I don't think I need to tell this audience, I guess, some of the other benefits of having genetic diagnosis in inherited retinal diseases. So one is that it does give us that diagnostic clarity and can inform better management. For a lot of people, it gives us information about family planning, if that's something you're planning to do. And also, knowing the genetic diagnosis can assist with access to gene therapies, because how do we know what gene to treat if we don't know what gene is causing the disease? But one other really important aspect why having genetic diagnosis and information is important is that actually that process of identifying how a gene is linked to disease can actually help us learn more about that particular inherited retinal disease. And so this is how we uncover how the disease works and actually develop new targeted treatments. Because without the knowledge of how the disease works, and also having people with these different inherited retinal diseases be involved in research for us to learn about that, there really would be no development of new gene-specific treatments. And so now I'm really going to talk through the steps of, I guess, what happens when you do donate a sample for blood tests or get a genetic test. So in the first step instance, I'm going to talk about what happens when you have this done in a clinical laboratory to try and get a diagnosis. Many people who are listening may have had genetic testing to try and identify their genetic diagnosis. And so I'm going to sort of talk about how this process generally works. And so in the first step, a blood or saliva sample is sent to the laboratory, and that sample is sequenced by usually some kind of big instrument in the laboratory. Now, when the output comes, it doesn't just come through in a report. There's actually a lot of steps in between. So the first step is that we know that in each person, the genome actually has about 3 billion base pairs. So that gets aligned to what we call a reference genome to try and find different changes in there that might be different or spelling mistakes. However, in each person, we actually have about 5 million, over 5 million DNA changes. So how do we know which one is causing disease? And so that gets mapped to a reference genome. It goes through a series of processing steps to try and find, map, and then filter all of these potential spelling mistakes that are in the DNA. And then most of the time, there is a person sitting in there who then interprets that data. And their goal is to assess the evidence, to use advanced tools to try and predict what all of these changes mean. But also another important aspect of what they do is look for other unrelated cases. And generally they have to be cases that have already been published in the literature. And so as generally, when you get diagnostic testing in a clinical laboratory, a confirmed genetic diagnosis or one that can be reported can only be found and reported if generally has to fit two criteria. So one, there has already been strong evidence linking that particular change or variant to an inherited retinal disease. And number two, someone else with the exact same change, not just in the same gene, but the exact same spelling mistake, has already been reported in the literature or a database, and they've presented with a similar disease, such that the researchers or the clinicians can now say definitively or with confidence, this, of the 5 million changes, this is the one that's associated or that is causing the inherited retinal disease. And only if both of these criteria are met can we then report a genetic finding in the genetic report. And so this is the case for about 50 to 60% of cases. And so if you've had a genetic test and you had a variant reported and a genetic diagnosis, what that means is that there was evidence that we found the change in the DNA. But what happens if a genetic cause is not found? And that's for about 40% of the cases, people who do get tested, that is the case. And if a genetic cause is not found, there could be several reasons. One is that it could be because of test limitations. So technology to this day is still developing, but only gets us so far. And some of the changes in the DNA might be really hard to detect, or it may not have been covered in the initial sequencing technique that was used. A second reason is that there could be unknown DNA changes identified. So a genetic change that may cause disease may have been identified, but that particular change is not quite well understood yet. And so what that means is that there's not enough evidence for us to associate that change with the inherited retinal disease. And so we can't say with confidence, this is a change that causes disease. And the third reason, and it's less likely now, could be that there's missing genes. Not in the person being tested, but overall, some of the changes just haven't quite been linked to some genes yet, and we still don't know about a number of genes that may or may not be causing disease. And so if that's the case, what we can generally do is one, often we can go back and retest using a different technique, but this is only beneficial for some people. Because if you imagine if it's because of the second reason that the change has already been found, but we don't have enough evidence associating with the disease yet, then going back and retesting using a different technique isn't necessarily going to be beneficial because we would just find the same change over and over again without generating any more evidence. Other things that we can do to generate more evidence to find out what these genetic changes mean is that sometimes we can involve more members or more family members to try and identify the inheritance of these changes. Sometimes we can conduct further experiments, so possibly stem cells or growing cell taking tissue cultures, so skin biopsies or blood cultures to try and grow cells and try and identify what is the cause, what does this genetic change actually do to the cell, the rods and cones. And sometimes we can collaborate internationally and nationally to try and find other families with the same spelling mistake who have the same inherited retinal disease, because that then generates evidence that this is a disease causing change. And so that is actually some of the work that we're doing starting this year. So we're very fortunate this year to be supported by the Advanced Genomics Collaboration Platform in Victoria, so at the University of Melbourne. And that's a partnership between the University of Melbourne and Illumina. And so we have a new project, and our goal is to establish a platform to try and find a diagnosis for people who could not get a diagnosis for their inherited retinal disease previously, and then to use this information to try and better test and diagnose inherited retinal diseases in the future and get people into emerging treatments. And so what we have been doing is we've invited people who have not had a diagnosis to attend a research eye test. And this is where we sort of will take images of the eye and learn more about their family and their eye history. This is really important because it allows us to learn more about the disease traits. And that's important information for understanding how the disease presents and might progress. And without that clinical data to match with the genetic data, it can be hard to connect if a certain genetic change is actually the cause of the condition. And the second thing is we're doing is then collecting a blood sample, and we're sending that away for a technique called genome sequencing. And so in this technique, what we're doing is sequencing the whole genome, and so we're broadening the search to a person's entire DNA profile. And so that's like, instead of flipping through certain pages of the book, we're actually just taking the whole library. And that gives us a comprehensive map of all of the genetic changes, including those that may not be well understood or previously have not been associated with inherited retinal diseases yet. And so I guess what does that actually look like, right? So when we do sequence the genome, we actually generate a lot of data. How much data is that? I'm going to use a few analogies now. And so on the screen, I've got a quote about how much data is a person's DNA. And so in each person, we each have 3 billion base pairs or 3 billion letters. And so if we printed all of our DNA, which is 3 billion letters on books, it would fill 4,000 books. Sorry, my husband's making a coffee in the background, so hopefully you can't hear that, but I can hear the coffee machine. And so 4,000 books, that is a lot of books in each person that you then have to wade through to try and find this one spelling mistake that might be associated with an inherited retinal disease. How do we do that? Well, I've got another analogy, and this one I actually took from Melbourne Genomics and adapted. And so that process, that genetic sequencing, it's like putting the 4,000 books through a shredder. And what we have to do is reassemble each book letter by letter. We then check every word for spelling mistakes. We then check every mistake to see if it alters the meaning of the sentence. We then check if that sentence changes the meaning of the paragraph. And we do this we do this by filtering through all of the significant changes or the significant mistakes that there might be in this whole library of the genome to try and find that one change in the 3 billion base pairs that might be causing disease. And it is a long and quite a tedious process, but it is the only way to find these new missing disease-causing diagnoses and changes. Along the way, there's a lot of other challenges and considerations. So one of the things is definitely that balance between privacy and data sharing. Because what really is the most important to us is that we know that genetic information is highly personal, and so protecting this data is our first priority. And so generally, even when we sequence any genome, it gets de-identified and anonymized even before it goes to the lab when it's done for research. However, another side of that is that considering that sharing some anonymized data with other researchers and genetic research is almost critical for finding other families with the same disease. Because if you imagine if every team just holds on to their own pool of patients and there's no that sharing of knowledge or information across, then we're never going to find someone else that might have the same disease causing change in another cohort because we don't know about them. And so this data sharing helps us connect the dots across families. And for rare diseases is particularly important because sharing that any sort of anonymized information can help us find patients in other studies in other countries to confirm or define other cases that might have the same disease. And not sharing does actually limit capacity to find a diagnosis. And so that's a balance that we really have to consider in any genetic research. And what we do in our study is after anonymizing all of the samples and sequencing, we then look for all the changes in our team first, look for any potential disease identifying changes, and we filter all of the data. And then we reach out to other databases. We take a subset of all of that anonymized data, and then we reach out to other databases, mostly around Australia at the moment, to try and go, hey, we've got this undiagnosed case with this particular change that we think is causing disease. Do you have anything similar? And we're doing that firstly across Australia. So we're establishing collaboration with the Australian Inherited Retinal Disease and DNA Bank team, which is a wonderful team over in Perth, so that we can work together collaboratively and solve more cases. And also then outside of eye diseases, we're also setting up paperwork with the Centre for Population Genomics, which is an initiative in rare diseases more broadly across Australia, led by Murdoch and Garvan Research Institutes. And really the idea is hopefully by collaborating with all of these national institutions and programs, while still protecting our patients and our data, we can help solve more diagnoses this way. And so looking more broadly now, aside from our study, are there other examples in the world where they have successfully used this type of analysis to try and find more diagnoses? In other words, can we learn from other countries and other studies around the world? And so I'm going to take you on a step back, and on the screen I'm showing a map of the world and different colors that really have colored different countries colored that have large biobanks. I'm going to drink of water. The countries with large biobanks that have facilitated genetic studies. And so the one that I'm really going to talk about is the UK biobank, because they're probably one of the most successful examples that have used gas data sharing to try and facilitate more diagnoses for rare diseases. And so the UK, they really offer genome sequencing to all patients in the national healthcare systems of the NHS through the 100,000 Genomes Project. And this was launched 10 years ago in 2012. And so in this research study, what they did is that they took 100,000 genomes from people with cancer and rare diseases, and they then they sequenced them in order to try and make more diagnoses, but also then promote scientific discovery through the integration of that information into healthcare and data sharing. And has that been successful? Well, that program then resulted in genome sequencing being incorporated as a part of the NHS in the UK. And so anyone who does go and through part of normal healthcare, public healthcare, can have access now to genome sequencing if they've got an eligible condition. And so this is all because of the 100,000, genomes and 100,000 people in the UK that have donated their data to research in an anonymised format to try and enable more diagnoses. In Australia, we don't quite have any infrastructure like that at the moment. And so currently we don't really have any national biobanks or data banks or that national infrastructure for trying to identify and sequence inherited retinal diseases. However, we are working towards trying to find more diagnoses still. Some of the challenges, again, with not having these bigger programs and bigger infrastructure is that then usually it means that programs are smaller. So we do run on limited funding. Usually we do run on smaller research grants. And what that means is that we are only able to sample a smaller number of cases, usually sort of tens or hundreds if we're lucky. And we do have fewer hands on deck to help analyze that data. But it is something that we're working towards, hopefully building our platform, building our research structure, and building our research facilities so we can try and find more diagnoses for more families with different inherited retinal diseases. What it does mean with smaller scale is that genetic analyses can be quite a lengthy process. And so sometimes it can take months or years for people to try and find a diagnosis. And a part of that is because of what I mentioned, is that it involves not only finding other families with the same spelling mistake, but also then enrolling them in research as well, so that we can try and build the evidence that's needed for confirmation. And so the progress can be slower with limited resources, but we are working on building our local capacity. But we do know that every single research participant does help accelerate that discovery to try and hopefully make future diagnoses faster and also more accurate as well. And so as a summary, I'm going to talk about three main points that I sort of mentioned. One is that we know that genetic discovery is resource intensive. It does generate a lot of data, particularly when we're looking for new diagnoses that were not previously reported. Second is that the results are only reported when we have strong evidence and when we have confidence that this particular spelling mistake or change of the 5 million is the one that's causing disease. Because what we don't want to do is give someone the wrong diagnosis. And number three is that building that strong evidence does take time. And it requires not only a lot of sequencing, a lot of laboratory studies, but also finding other families with the same condition. And so sometimes that can take a little bit of time. And importantly, a genetic diagnosis isn't just about finding that change in the DNA, but really making sure that the change is the one that's causing the inherited retinal disease. What's next for us is that I've talked a little more, and Sally mentioned at the start that we have a big data-based registry. We really would love to connect with anyone else or anyone who's interested in hearing more about our work or being a part of our research. And that's very much, we don't see our research as a siloed process. I see this as that we are really collaborating with families with inherited retinal diseases, with people who are interested in contributing to work together to try and find more diagnoses and also to get people into treatments for their conditions as well. And so what I'm giving is my time and my brain, but very much we see this as a collaborative process, because without the involvement of families that have inherited retinal diseases, without hearing about what it is that they really want to hear, then we probably wouldn't be in this field of research. And so if you are interested in connecting with us, if you haven't already, the information about our registry is on the Retina Australia website. So that's the Venture study. You're also always welcome to email us as well. And we have a group email. Again, this is on the Retina Australia website, but it's ird@groups.unimelb.edu.au. Or you can give us a call on 038344 3441. This should be on the website as well, and leave a message. It does go straight to a voicemail, so it goes to Lauren's voicemail, but we will give you a call back when one of our team members has a chance. And our venture study really is, I guess, that big registry and the initial process where what we use to contact people about different research studies. And so that might be clinical studies, inviting people to come in, clinical trials, if there is one running at the time, patient voice studies, so survey perspectives and hearing sort of interview type of studies as well, and genetic studies as well. And so generally as a first year, we always contact people through our venture study. And so lastly, I really want to finish off by telling you a little more about some of the team who, if you do contact us or if you have contacted us in the past, who you'll be hearing from. And so on the screen, I have a picture of all of the team, of our entire team. Well, it actually was taken 3 years ago on the picture on the left at our Christmas picnic. So it's not actually our entire team. There's been some movement, but on the right of the slide, I've got some of the people that have been really directly involved in some of the research that I do in the genetic testing and venture registry as well. So on the top left, that's Dr. Tom Edwards, who's a vitreoretinal surgeon, and next to him, second on the left, is Associate Professor Lauren Ayton, whom the two of them co-lead our overall program. Third on the top row on the left, that's Justin Dennis, who's the study coordinator on our genomic sequencing studies. And top right is Dr. Samudra Barasekera, who does a lot of the data analysis of the genetic data with myself. On the bottom left is Janice Hermawan, and she is the lead coordinator on our venture registry, whom hopefully some of you may have heard from in the past. Janice is actually going on maternity leave at the end of the year, and some of her role will be taken over by Elise Chicello, who's second on the bottom left. And Elise is going to be a study coordinator on the venture registry. Elise also leads a lot of the other industry studies at the Centre for Eye Research Australia as well, and including clinical trials. And lastly, third on the bottom row is Parker Trong, who's a genetic counsellor in our team. And bottom right is Sujani Thrimavasana, and Sujani is an study optometrist and also coordinator who helps with a lot of the registrations and also processing referrals and triages as well. All right, and thanks very much.
Thank you so much, Ceecee. That was fantastic. It's so interesting to hear about the project and all the work that you're doing, and to hear about what's happening overseas as well. Obviously, there's just a great deal of stuff happening. I'd like to introduce our final speaker for today, Dino Farronato, who is a Retina Australia board member. Dino faces the challenges of dealing with an inherited retinal disease on a daily basis. He was diagnosed with retinitis pigmentosa in his early 30s and is constantly adapting to consequential changes to his vision. He is committed to supporting others affected by inherited retinal diseases, as well as raising funds required to provide grants for innovative research into such diseases. Dino will briefly discuss his involvement in the venture clinical and genetic study conducted by CERA. He will cover his motivation for joining the study, his experience as a participant, and the follow-up once the genetic testing was completed. Thanks so much, Dino.
Thank you, Sally, for those that kind introduction. It's an honour for me to be part of this webinar presentation organised by Retina Australia, and to be part of the same program as Anai and Ceecee. I'm in awe of their great brains and all the great work that they do in this area of research. I really bring the patient perspective to this discussion, and I have a very simple and straightforward message. And that is, if there is an opportunity for you to be involved in research projects or clinical trials, I strongly encourage you to do that. I thought I would flesh out that simple message by retelling some of my own experience. So I'm a lawyer, a company director, a husband, a father, a keen bushwalker, and a rugby union tragic. I also have low vision because, as Sally mentioned in her introduction, I have retinitis pigmentosa. So I am slowly going blind. My introduction to the world of RP was when I was first diagnosed in my 30s. I went to the ophthalmologist because my vision was declining, and it was the classic good news, bad news story. He said, you've got cataracts. We can fix those with some intraocular lenses. But unfortunately, there's nothing we can do about the RP. And to which my response was, the R what? It was my first introduction to this disease that has significantly impacted my life throughout. Ever since that time, I've been quite enthusiastic about trying to do my own research. I've travelled to various medical facilities, trying to find what the current status of research, whether there were any things that could be done to minimise the impact of RP. At first, it was quite heartening because I was constantly being told there is nothing at the moment that is anything like a cure or a treatment, but keep your eyes in good order, because within 5 to 10 years, we're very confident there is going to be something. The one constant thing is that 5 to 10 year time frame for something to come up has been throughout those 30 years. It's just constantly been rolling forward. However, with everything that's been going on now, and I mean, you just have to listen to the work that Anai is talking about with organoids and gene therapy and stem cells, you can't help but feel really quite excited about the future for people with RP. If it's not my generation, then certainly the next one is going to have so much more support than I have had, really. So, I mean, there's lots of reasons why you should be participating in research projects and clinical trials. Obviously, there's the very self-interested one of, if you're involved in research projects and there is a possibility of a clinical trial which will assist you, then that is a great way in rather than waiting for the back of the queue. The second is a more altruistic point of view, of really just giving the researchers like Anai and Ceecee the tools that they need, the data, to really advance the research that's being undertaken. And the third one that was certainly very important to me was really that process of self-education, finding out about this disease, finding out about what's happening, and not living in that world of fear, basically, and an unknown. I feel much more enthusiastic and upbeat about the future, knowing what's happening with medical research. I think the way forward is looking positive, and I think there's a great advantage to everyone from participating in research projects and clinical trials. If not for yourselves and for those others around you, then for the next generation. So with that, I'd like to again thank Anai and Ceecee for all the work that they do. I know that there are plenty of ways in which you can find information about research projects and clinical trials. The one I recommend is if you go through the Retina Australia website, and there is a register there of trials. It's a great source of information for you. So again, repeat my simple and clear message: get involved, make a difference. Thank you.
Thank you so much to Dino, and thank you to all our presenters this afternoon. Now we have time for some questions.
Sally, can I dive in with a question? This is Dino Farronato.
Sure.
Renee, when can I, bearing in mind that discussion I was saying about 5 to 10 years, when can I be looking to get some stem cells injected into my eyes?
Thank you, Dino. We always put on the spot like that. Look, we have large MRFF, so that's a medical research funding grant at the moment, that it's a 5 years project to, again, as I explained, change all the ingredients of what we are doing in the normal lab so that we can go into manufacturing. So this it's what we envisage for this process. 5 years we had all the ingredients ready. And then, of course, what we need to do next is to go into a facility that can manufacture. And that's going to be perhaps another 2-3 years, working out the manufacturing of these cells and having the cells ready to go into patients. Meanwhile, we need to be doing large animal studies to do tox studies to show that these cells will be safe. And then, meanwhile, we'll be applying for funding to do the clinical trials, which is not trivial. So there's a lot of, what I'm trying to say, there's a lot of balls that need to fall in place so that we can move to the clinics. And it's so difficult to give a next number of years. Of course, it's very exciting because we are moving towards it. Just the fact that we have this funding to be able to, manufacture the cells and try and do everything that we have to do to transplant the cells into patients is just an unknown. And even having this pathway so clear, we're not sure if we are going to get funding, if we are going to move forward at the end of the 5 years because of that unknown. We'll try our best as always. And yeah, I guess it's just difficult to predict. But it's happening.
I'm here 5 to 10 years.
Look, it's funny because these numbers always appear, right? But I just came back from a conference in an international conference, and the researchers were saying 5 years ago we were sitting in this room and there wasn't any clinical trials for Parkinson's disease, and now I was seeing results of 3 clinical trials showing very safe stem cell derived treatments for Parkinson's and some efficacy. So it was remarkable to see that those researchers presenting and working together, actually, to all working on very similar projects, transplantation of cells of neurons in the brain, but working together to move the technology. And I think, and there was actually a lot happening in the eye as well with RPE cells. As you might know, the transplantation of retinal pigmented epithelial cells has happened in patients. That were clinical trials, and a few clinical trials is still happening. We, of course, have a bit of a harder task because to produce photoreceptor cells is a bit more laborious and takes a bit more time. So that's why the photoreceptors transplantation field is still catching up. But the message, I came back from this conference with really thinking very positive about the field, because it has evolved so quickly that that's why I say we're getting there, and it's very exciting.
Thank you for your work.
Thank you.
That's great. I've got a question for you, Anai, and I think I ask this question every time, but I'm just sort of fascinated by it. When you're introducing the artificial or the laboratory grown cells into the affected eye, am I right in thinking that there's no immune function there? So there's not going to be any rejection of that tissue, unlike transplanting a kidney or something.
Yeah, so the eye, it's relatively more protected than other organs. So the brain and the eye actually are relatively more protected because we have this blood-brain barrier that makes the environment a bit more protected from immune rejections and from the attack of immune cells. That is not to say that that doesn't happen. And one of the things that the field in general has worked out is that if patients will be receiving human cells, for example, for the RPE transplantations that happen in patients, the patient will have a 6 months treatment with immune suppressants.
Right.
So that will happen, and it's most likely it's going to start a few weeks before the therapy and then last for 6 months. So in the brain, it lasts for a year. In the eye, 6 months. So it's again very interesting to know that we are already, we know these regimens, we know what we need to give, and of course for pancreas transplantation, for example, we just had some really nice data coming out of clinical trials with islet cells for pancreas for diabetes. They will go on immunosuppressants throughout life. So yes, I think in the eye, it is a better scenario.
That's better. That's great. Thank you so much. I think we have time for one more question, and that is from Stephen Miners, who is in the room with Dino. And so, Stephen, I don't need to do anything to your microphone. You can just ask your question, I think.
So hopefully you can hear me. So look, thank you very much for all the presenters. As someone who's just getting my head around some of this, my head has both exploded, but there's some absolutely brilliant stuff going on. So thank you all for that work. My question though is for Ceecee. I'm just wondering to what extent AI is actually making your job easier, because it looks more like a data processing issue than anything else in a lot of ways, or whether you're still actually held back by other things like privacy, getting a database, being able to do comparative stuff. It's all very well to test someone's DNA quicker, but if you can't get a comparison to find common themes, whether that's what's actually holding us back. It'd be interesting in your view of how much AI can take you forward over the next few years.
Yeah, I think I've got the same question as well, Stephen. So I think AI, if we think about what AI does, is that it takes a lot of data and it helps process faster, right? At least if anyone in the audience has more knowledge about AI than I do, then do feel free to pipe in and tell them I'm wrong, but that's what AI does. And so what it doesn't help us do is generate new evidence. And so it can certainly help us facilitate some of the processing of data. So for example, we're actually trialing a new AI tool with Illumina as a part of our processing data to help try and filter the 5 million variants faster in each individual. And so what we're doing is seeing, okay, can the AI, save us 5 hours of work, which, if you think about it per person, is actually quite a lot in big diagnostic settings. But even then, because I do think that in this field, you need someone to validate the AI. You need someone to really go, is this actually the right one? Is there evidence for this? We still do require a person to sort of sit there and go, right, this is wrong. No, we can't report that. That is the step. And so I think overall, I would say I certainly will be very interested to see how AI can help us in the next 5 years in the field of genomics. What it can do is hopefully save us some of that processing time, make processing that data a little bit faster. What I don't see it doing for us yet is finding those other families around the world who do have the same variants. That will still rely on all of us to encourage people to be a part of our research, and also to talk to other researchers around the world as well, but also then to build the infrastructure to make sure that the data can be shared safely. But yeah, maybe I'll do another talk in 5 years and maybe my answer will be completely different. So.
Yeah. Thank you.
Fantastic. All right, thank you very much to all of our speakers today. Anai, Ceecee, and Dino. And thank you to all the participants for your attendance. I'd just like to hand over to Leighton Boyd now just to say a few words.
Thank you, Sally. Look, it's always amazing in all the years that I've been involved to have the quality of work that's going on. I mean, I've been fortunate to know Anai for quite a long time. I've been fortunate enough to go through the lab up there. It's just one of the most amazing visits that I've ever had in my time with Retina Australia. To see those organoids and the actions that are going on there, it's just astounding. And thank you, Anai, for all your work. It's been amazing, and we're just so grateful, I think, as patients, people that are, really wanting to get some sort of treatment or cure down the track, even if it's not for us, our children or grandchildren or other generations. So thank you and your team. It's amazing work that's happening there. Ceecee, that work that you people in your labs, the venture studies, I don't know how many questions I've asked for your team, but there seems to be lots and lots and lots, but it is really important work because us who do have an IRD, it's really important that, as Dino said, that we do help out with the researchers, know what we have, what sort of condition, what sort of IRD, and then give out the next generation of people the opportunity to work on a cure. And us on the board of Retina Australia, we're really lucky to have Dino. Now, Dino, he's stepping here with us at Retina Australia. He brings his professional knowledge. He also brings his lived experience. So I do encourage people to get involved with not-for-profits, Retina Australia or other groups, just so it can help us do the work that we do. So I'd like to thank the three of you on behalf of all of us and the organisation for your contribution today. And also thank Julia and Sally for putting this event through. This has been an amazing series this year, and I thank you all for your work. And just on a final note, for those who don't know, Sally is actually going to retire. I don't know whether that's allowed, but she is going to retire. But enjoy your retirement, Sally. We've certainly enjoyed your time with Retina Australia, and I hope you have many happy, long years. Thank you. I'll hand back to you, Sally.
Thanks, Leighton. I hope I have many happy, long years too. Okay, so that's the end of the session now. I hope everybody's found it very informative. If you still have any questions, please let us know and we will endeavour to put you in touch with someone who can help you. So that is the end of the webinar. Thank you all very much. Thank you to all speakers and to everybody for coming, and enjoy the rest of your day.